Hello, everybody. Good evening to you all. Hello, everyone. How are you all doing today? So, I'm happy to see you all here waiting. Yes. Can I have some quick highs from all of you? Yeah. Good evening, Rohit. Good evening, Jasmine. All the joinees, can I have some quick hellos with you all? Hi, Disha. Okay, so I hope all of you are doing good. So today we are going to solve the genetic mystery of extensions of Mendelian principles. So if you remember in the last live class that we had day before yesterday, we talked about Mendelian principles. So I did not go in the basics or the topics of Mendelian principles. We started attacking the part C questions, right? Because name of our series is Prahar. So we want to attack, right? We want to attack the part C questions so that in next CSIR exams, they will not run away from us, right? Okay, so now today we are going to talk about the extensions of Mendelian principles. So are you all ready for that? Are you all ready? Can I have some quick BIOs? Come on, everyone. Be active. I want all of you to solve the questions with me. It's not only me solving it. सबको साथ में solve करना है, right? Okay, so what I will do is I'll turn off my camera so that we can have an uninterrupted streaming. Okay, and then we will start looking at the questions. Okay, so turning off my camera does not mean I'm not here, I'm right here. And we will solve the questions. All right. Okay, so starting with the very first question, which is up. Okay, so this is one of the previous year question, part C. So this is the pathway kind of question. Okay, so I told you in the last class also that we'll be handling some pathway kind of questions. Okay, so this pathway questions, I'll tell you that they are very easy actually. Okay, but again, just looking at the pathways, just seeing that I don't know this, how will I solve it? Most of you will not even touch these questions. So let's do it together and I'll show you how it can be done very easily. Good evening, Ayushi. Good evening. So you are on time. We are just starting the question discussion. So hypothetical biochemical pathway for the formation of eye color in insects is given below. Now, two autosomal recessive mutants, small a and small b, are identified which block the pathway as shown above. Okay. Considering that the mutants are not linked, what will be the phenotype of the F2 progeny if crosses were made between parents of the genotype this and this, and the F1 progeny are intercrossed? Okay, so let's see the pathway first. Now, very clearly, the pathway is saying, yeah, good evening, Biotechy, Priyadarshini. Okay, so what is the pathway saying here? Pathway is saying that we have one substrate X which is colorless. It is converted into a colorless intermediate, and that colorless intermediate is acted upon by the capital A allele. See, it is written what? That if you have a mutant small a allele, then this pathway is crossed. Okay, that means it cannot happen. So when will this conversion happen? Conversion will happen if the alternate allele, that is capital A, is present. Now, so if small a is present, then this brown pigment is not formed. But if capital A is present, then the brown pigment will be formed. Do you people understand this much? Now, in substrate Y, what it is showing? That substrate Y, it can form the colorless intermediate, but if mutant small b is present, it cannot form. Okay. So again, what does that mean? What is the alternate thing? That is not mentioned in the question, but you have to understand. That means the alternate allele, that if it is capital B allele, then this conversion will happen. And then the colorless intermediate can also be converted into the orange pigment. And if both brown and orange pigments are present, then the eye color will be wild, which is orange brown. Okay. Now, what is the cross talking? So cross is saying that small a, capital B is crossed with capital A, small b. Okay. So this is our parental generation. Then we will form the gametes here. Gamete will be small a, capital B, only one type because individual is homozygous. Here also the individual is homozygous, so only one type of gamete, capital A, small b. Okay. And then the F1 generation will be formed. So F1 will just be a fusion of these two. So it will be capital A, capital B, small b. Okay. Next, what it is saying, F1 progeny is intercrossed. Means you are taking two F1s and crossing them. So what will be the F1 intercross or F1 self-cross that we write it as? So the cross will be this. Okay. Now, this is a dihybrid cross. Correct? So in the dihybrid cross, what is the expected ratio? What is the expected phenotypic ratio of dihybrid cross? Quickly. What is the dihybrid ratio of a phenotypic ratio of a dihybrid cross? It is 9:3:3:1. Come on, guys. Are you not revising? What happened? Nobody answered. ठीक है. So 9:3:3:1. What is it? Okay. So 9/16 is the proportion of what? 9/16 is the proportion where both capital A and capital B are present. Okay. Both the genes are showing their dominant expression. That is what this 9/16 proportion is. Okay. जिसमें दोनों dominant allele express हो रहे हैं. Right? So I'm putting a dash here because any of the allele can come and take up the place. It can be either small a or it can be small b. So any allele can come and take up the place. That's why I'm putting a blank here. Okay. Next, what is the next ratio? 3/16. What is this 3/16 ratio? It is for one dominant and one recessive. Okay. एक dominant है, एक recessive है. तो हम मान लेते हैं कि यहाँ पे A जो है वो dominant expression हो रहा है और B का recessive है. ठीक है. Other 3/16 will be again one recessive, just the opposite. So one recessive and one dominant. So यहाँ पे A recessive मान लेते हैं और B है dominant. और 1/16 क्या होगा? जहां पे दोनों recessive है. Correct? अब हम इसमें से देखते हैं, अब हमको पाथवे देखना है, ठीक है। अब हमको पाथवे देख के डिसाइड करना है कि कौन सा कलर फॉर्म होगा. सो वी हैव टू चेक द पाथवे नाउ एंड सी विच phenotype will be formed. Right? Now, in the first scenario, what do we have here? So, capital A भी है और capital B भी है. So, if capital A है, means brown pigment तक form होगा. Capital B है, means orange pigment तक form होगा. Or brown and orange both are present, means the wild type eye color will be formed. Okay. So, this 9/16 will be which eye color? Orange brown. समझ में आ रहा है सबको? अब ये 3/16 जो है उसमें A कैपिटल है, मींस ये वाला फॉर्मेशन तो हो रहा है, दैट इज द ब्राउन कलर इज गेटिंग फॉर्म्ड, बट यहाँ पे स्मॉल बी है. सो स्मॉल बी मींस ये पाथवे यहाँ पे रुक गया. ओके यहाँ पे जो वाई सबस्ट्रेट का पाथवे है इन सेकेंड सिनेरियो वो नहीं हो रहा है, सिर्फ ब्राउन पिगमेंट फॉर्म होगा. ओके सो ये वाला जो जीनोटाइप है विच इज कैपिटल A डैश स्मॉल बी is going to form the brown pigment only. Right? The other proportion, that is 3/16, it is small a capital B dash. So if small a is there, means अब यहाँ पे ये वाला pathway जो है वो रुक गया है. All right. So what I will do is I'll just use it color. So this second one, let's say, is blue. So this pathway is happening. Okay. For the next one, let me use green. So for that, this pathway is blocked because it is small a. Okay, so this pathway has got blocked, but this pathway, the Y substrate का जो pathway है वो हो रहा है, तो यहाँ पे हो जाएगा orange color. Right? And finally, when it is small a, then none of it will be working. यहाँ पे भी cross हो गया क्योंकि capital A है नहीं, और यहाँ पे भी cross हो गया क्योंकि capital B है नहीं. Okay, so colorless ही रह जाएगा. तो यहाँ पे small a जो genotype है उसका क्या phenotype होगा? What will be the phenotype? It will be colorless. Okay, because none of the intermediates could be formed. So what will be the ratio here? The ratio will be orange brown, that is 9, is to 3, which is brown, okay, is to 3, which is orange, is to 1, which will be colorless. Okay, so you will see that answer will be option B. Do you all understand? Okay, is it clear, everybody? All right. So if it is not understood, if you have any doubt, please write it down at the end of the class. As you saw in the last session, also we will have a personal Zoom meeting. Okay, so we can have a one-on-one doubt clearing session there. So please join that Zoom meeting. The link will be given at the end. Along with that, the link for some freebies that will help you in CSIR exam will also be given at the end of the session. So please remain till the end. Okay. So if you have any doubt, if you did not understand this solution, please write it down your question and we can take it up in the Zoom class. Okay. You can ask me personally and I will explain it to you. All right. Moving on to the second question. So the following is the biochemical pathway for purple pigment production in flowers of sweet pea. Okay. What is the pathway given? Pathway is that colorless precursor 1 is converted by allele capital A into colorless precursor 2. And that is converted by allele capital B into purple pigment. Okay. So recessive mutations of gene A, capital A and B leads to the formation of white flowers. Okay. So clearly it is mentioned that if it is a recessive mutation of any of these genes, then it will result in white flower. Okay, so we can modify the pathway that if it is small a, okay, if it is small a, right? Sorry, if it is small b here, okay, so this formation will not happen if it is small a. If it is small b, then this conversion will not happen. And in both the scenarios, it will form white flower color. Okay. So what can you understand from this pathway? So this pathway clearly shows that the purple flower color can only form if capital A and capital B are both present together. Right? Any one of them absent or both of them absent will result into white color. Right? Now, if you correlate it with the gene interactions, okay, so you will see that this phenomena is exactly same as the complementary gene action, or which we call as the duplicate recessive epistasis, isn't it? So where both the alleles are required for the formation of a particular character. So if any one of them are absent, both the genes are required for the formation of that particular character. If any one of them is absent, then that particular character will not be formed. Okay. So this is just like, this is the complementary gene action actually. So now the cross is given. Okay, so the cross is made between two parents of genotype this and this. Considering that the two genes are not linked, the phenotype of the expected progenies are. So let me write down the cross here. It will be better. So capital A small a, capital B small b is crossed with homozygous recessive. Right. Now in the next generation, what do we expect? So this individual is homozygous. It will form only one type of gamete, small a, small b. This individual is heterozygous. So it will form four different gametes. Capital A, capital B, capital A, small a, capital B, and small a, small b. Okay. Now in the next generation, okay, what will be the scenario? So you can make a Punnett square. So when we make a Punnett square like this, okay, so here let's put this AB, and here let's put the four different gametes that we have got. Okay, now अब देखते हैं कि यहाँ पे क्या क्या combination हो सकते हैं. So you see this all combinations will be possible. Okay, so you write down the possible combinations. ठीक है, तो जैसे भी हमारे पास pathway आता है ना, तो जो भी क्रॉस है पहले वो क्रॉस कर लीजिए, फिर उस क्रॉस में जो पॉसिबल जीनोटाइप्स है उसको पुनेट स्क्वायर में फाइंड इट आउट एंड देन यू चेक विद द पाथवे. Okay, so Pooja, you are saying 9:7. Well, the answer is not that. Okay, now I'll show you how. So here, see, it is homozygous recessive here. It is homozygous recessive. 9:1 is the ratio in the F2 generation of complementary gene action, but that you get when two heterozygotes are crossed. Is it not? So if the cross would have been ये वाला कि दोनों हेट्रोजाइगोट्स है और उनका क्रॉस हो रहा है, ठीक है, और कॉम्प्लीमेंट्री जीन एक्शन है, तब आपको 9:7 रेशियो मिलता. पर यहाँ पे क्या है, यहाँ पे होमोजाइगस रिसेसिव के साथ क्रॉस है, right? So that is why it becomes very important to see what is the genotype given. Okay, आपको पता है कॉम्प्लीमेंट्री जीन एक्शन, पर अगर आप हड़बड़ी में इसको आंसर कर देंगे तो it will be wrong. So now you have found out the genotype. Now let's see what are the possible, what will be the phenotype in them. Okay, so I'll write down the pathway. So if I'm not wrong, so there is precursor 1, which is colorless, is converted by capital A into another precursor 2, which is colorless, that is converted by capital B allele into purple flower, right? This is the pathway given. Okay, now that means what? So this individual, which is capital A and capital B, both are present. Okay, so what will be the color formed here? So it will convert P1 to P2 and P2 to purple. So the flower color here will be purple. Okay, now what about the next one? And we'll just choose blue. Okay, so in this scenario, capital A is there. So this conversion is possible. Okay, but it is small b, means this conversion is not possible. Okay, that means the flower color formed will be white. Correct? Colorless. Okay, colorless basically is meaning white. Okay, so you cannot have a colorless flower. It has to be white. Now, the second scenario, when it is small a. Okay, so small a means ये वाला जो conversion है P1 to P2 यहाँ पे ही नहीं हो रहा है. P1 to P2 conversion is only not happening because it is small a. So यहाँ पे capital B है, पर capital B can only work on P2 and P2 can be formed by capital A from P1. So if capital is not there, तो P2 बनेगा ही नहीं और P2 नहीं बनेगा तो capital B उसको पर्पल में कन्वर्ट कर ही नहीं सकता. ठीक है, तो इस सिनेरियो में भी हो जाएगा फ्लावर कलर इज व्हाइट. एंड द थर्ड सिनेरियो when both are recessive, so इसमें भी it will be again white only because again this conversion is not possible. Okay, so कितने white हो गए यहाँ पे 1 2 3 और पर्पल कितने हैं 1. So if you write down the ratio, it will be purple is to white. कितना होगा 1 purple is to 3 white. Okay, yes, whether you write it as 1 purple is to 3 white or you write it as 3 white is to 1 purple, it's the same thing. So let's check it out. Do we have any option? Yes, we have option B, that is 3 white to 1 purple. Okay, I hope all of you understood, right? So don't be in a hurry to answer. Yes, I know time management is important, but time management should be in the correct way. Okay, you just do not be in a hurry और फिर आता हुआ सवाल गलत हो जाएगा, है ना. ये बहुत easy था, पर आप अगर जल्दी में करोगे तो वो 9:7 कर दोगे तो it will be wrong. Okay, so it's important to see what cross it has given. All right, moving on to the third question. Okay, now this question is, it was given in the last CSIR exam only, and this is regarding the complementation test. Now, complementation test is a small topic which is also included in your the extension of Mendelian principle. Okay, so I hope all of you are aware with the complementation test. Are you? Are you all aware with the complementation test? Okay, Priyadarshini, what about others? ऐसा मत बोलना है कि पहली बार सुन रहे हैं. If you have prepared genetics, then for sure you must have studied about complementation test. So complementation test is a test which is conducted to find out if two mutants showing similar mutant phenotype, if they are having the mutation in the same gene or different gene, okay, without any sequencing, without any molecular analysis. Okay, so complementation test is basically a crossing method. Okay, we cross two individuals showing the mutant phenotype, or we cross two individuals who are homozygous for particular mutation. Okay, and then we try to find out whether in the next generation, in the next generation, what is the phenotype. On the basis of that, we try to establish whether the mutants are having a mutation in the same gene or in different gene. Okay, now let's see the question here. So a Drosophila mutant, that is line A, with vestigial wing is isolated in a laboratory. The vestigial wing phenotype was observed to be recessive and mapped onto gene X. 3 other laboratories also isolated vestigial mutants. Okay, so what do you have? You have mutants all having vestigial wings. Okay, so mutants having similar mutant phenotype. And they call these lines as B, C, and D. Okay, right? So now you want to check whether, so in order to test if the mutation in the lines B to D also mapped to gene X, the following crosses were made and the phenotype of F1 progeny was observed. Okay, so this is the cross. This column here shows the cross and this column here shows the morphology of the F1 progeny. Based on the above, identify the lines which is most likely not to have a mutation in gene X. ओके सी ये जो है, ये यहाँ पे आप देखोगे तो यहाँ पे क्वेश्चन में कहीं पे भी कॉम्प्लीमेंटेशन टेस्ट मेंशन ही नहीं है, ठीक है। पर हमको समझना पड़ेगा कि ये कॉम्प्लीमेंटेशन टेस्ट की ही बात हो रही है, बिकॉज वी आर ट्राइंग टू मैप वेदर two or more mutants having similar mutant phenotype, whether they are present on the same gene or not. So ये जो पूरा लॉजिक है, ठीक है, ये क्रॉस से पता करना कि दो म्यूटेशंस, दो सिमिलर म्यूटेशंस एक ही जीन में प्रेजेंट है और डिफरेंट जीन में प्रेजेंट है, वही कॉन्सेप्ट है कॉम्प्लीमेंटेशन टेस्ट का, ठीक है। तो यहाँ पे आपको समझना पड़ेगा दैट यू हैव टू अप्लाई द कॉन्सेप्ट ऑफ कॉम्प्लीमेंटेशन टेस्ट इवन दो इट इज नॉट मेंशन्ड हियर. Okay, and let me tell you one thing that earlier very few times question used to come from complementation test, पर अभी 2-3 बार से हम ऐसा देख रहे हैं कि हर बार ही complementation test का कोई ना कोई एक सवाल आ रहा है, ठीक है. It can be in part B और it can be in part C. ओके और फिर क्वेश्चंस ऐसे आ रहे हैं कि आपको पढ़ के समझना पड़ेगा कि यहाँ पे कॉम्प्लीमेंटेशन टेस्ट का लॉजिक अप्लाई करना है. दे विल नॉट राइट इट लाइक दैट. ओके सो हाउ टू सॉल्व इट नाउ? Now in complementation test, so the thing that you have to remember in complementation test is we cross mutants, okay, homozygous mutants. So if mutant is crossed with another mutant, right, and the result is mutant again, okay, mutant crossed with mutant, F1 is also mutant, okay, that means no complementation happened. No complementation, okay. And when will no complementation happen? When the mutations are in the same gene, okay. So see, complementation अगर हम कहते हैं तो complementation is the process of making something complete, right? तो अगर हम जैसे अगर मीठा खाते हैं तो रबड़ी जलेबी का एक बहुत बढ़िया कॉम्बिनेशन बनता है, राइट? रबड़ी क्रिस्पी, हॉट and sorry jalebi is crispy and hot, whereas rabdi will be soft, creamy, right? So ये जो दोनों हम जब मिला के खाते हैं तो वो बहुत अच्छा कॉम्बिनेशन होता है, राइट? बिकॉज दे आर डिफरेंट. अगर आप दो जलेबियां साथ में खाएंगे या सिर्फ रबड़ी रबड़ी साथ में खाएंगे तो वो मजा नहीं आएगा. ओके सो म्यूटेंट क्रॉस म्यूटेंट, if the F1 is also a mutant, okay, that means there is no complementation happened, okay. And if no complementation means both the mutations that you were checking happened in the same gene, ये दोनों mutations एक ही gene में हुए थे, तभी यहाँ पे F1 में कोई फर्क ही नहीं पड़ा, it is still a mutant, okay. No complementation, so mutation is in same gene, okay. Now the other situation, when you cross mutant and mutant, okay, and in F1 you got wild type, ठीक है, जैसे रबड़ी और जलेबी को मिला के खा लिया और मजा आ गया, ठीक है, बिकॉज दे वर ऑफ टू डिफरेंट टेक्सचर्स, टू डिफरेंट quality, okay. So then this situation, when you cross two mutants and F1 is wild type, that is when we say that complementation has happened, okay. And complementation कब होगा? जब both the mutations are occurring in different gene, okay. So complementation test में ये जो basic thing है ना, ये आपको याद रखनी पड़ेगी. When mutant cross mutant, if F1 is mutant, that means both the mutations happened in the same gene, so no complementation. Mutant cross mutant, if F1 is wild type, means complementation happened, and that will be the situation when mutation was in the different gene, okay. Now see, vestigial is mentioned as the mutant phenotype, okay. So wild type phenotype क्या होगा? Definitely normal, right? So अब ये क्रॉस देखते हैं. A cross B is vestigial. So A and B means what? What will be our conclusion for this? A cross B is vestigial. Quickly can I have some answer? What will be the conclusion here? A and B, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different gene? A and B's mutation, are they in the same gene or different g Yes, that is correct. It is in the same gene. क्यों same gene? क्योंकि mutant को mutant से cross किया और F1 का फिर से mutant phenotype ही आया. Okay, that means A, B are in same. Okay, A and C vestigial again means same. Okay, now A and D when you are crossing, then you are getting normal, which is the wild type, right? Which means it has happened in different genes. Okay. Again, B and C vestigial means B and C are same. Okay. B and D is normal means it is indifferent, and C and D means it is indifferent. Okay. So now if we have to make two groups, right? So what do you see here? That AB if you cross, same. AC cross, same. BC cross, same. Okay. That means A, B and C, they all are happening in the same gene. Okay. But when you cross A with D or B with D or C with D, then it is different. Means C is a different gene. So if this is gene 1, so it is already mentioned, right? It is gene X. So if A, B, C are the mutations happening in gene X, then D is happening in some other gene. Let's say it is gene number 2. Okay. So the question based on the above, identify the line which is most likely not to have a mutation in gene X. So it will be line D only. Option number 3 or C. Okay. So I hope all of you understood this. Right. Now moving on to next question. Now this question was also there in previous CSIR exam, just the CSIR exam that happened last time. So that was about polydactyly. Okay. So it was the human polydactyly traits having extra finger or toes are caused by a dominant allele. In a screening, it was found that out of the 42 individuals having an allele for polydactyly, only 38 of them were polydactylous. Which of the following is the correct interpretation of the observation? Okay, so polydactyly means it's the gene that responsible for having more than 5 digits in hand and toes. Okay, more than 5 fingers or more than 5 toes if you have, so that is because of this situation which is called as polydactyly. Okay, now polydactyly is caused by a dominant allele. Okay, now, if it is a dominant allele, what is its feature? That even a single allele present, even a single allele will produce the phenotype. Okay, even a single allele present will produce the phenotype, right? Now, in the screening, it was found that out of 42 individuals having an allele for polydactyly, 38 of them were polydactylous. Okay, so you screened 42 individuals. Okay, so you found that all 42 were having all 42 individuals, they were having one allele for polydactyly. Okay, but but of them only 38 were actually polydactylous. Okay, they only, so 42 of them were having the allele, causative allele, but only 38 of them were having the extra digit. So what is the correct interpretation? So here actually means what? That you have in this group around 4 people who, even though they have the allele responsible for the extra digit, but they do not have it. Okay, they are not actually showing the extra digit, and that is because of penetrance. That is because of the phenomena called as penetrance. Right. So penetrance basically talks about the situation where so penetrance is decided by the number of individuals who are showing a particular phenotype for which they have the genotype. Okay, so 42 people, they have the genotype to be polydactylous, but only 38 out of them is showing. Okay, so what will be the penetrance in this situation? So this penetrance will be, the penetrance will be 38 divided, so it will be out of 42. So you have 42 individuals, out of that only 38 are showing polydactylous. Okay, so penetrance will be equal to 38 by 42. Okay, and in terms of percentage, if you have to convert, so multiply it with 100, so you will get 90% as the penetrance. Okay, so what will be the correct interpretation? The answer will be option A, that is the penetrance of polydactyly is estimated to be 90%. All right, I hope all of you understood this. Now moving on to the next question. Okay, so this is talking about another phenomena which was not observed by Mendel. Okay, so this is in some sheep, horns are produced by an autosomal allele, capital H, which is dominant in males and recessive in females. H plus individuals are hornless. Dominant in males, recessive in females. Okay, this allele. The horned female is crossed with a hornless male. One of the resulting F1 females is crossed with a hornless male. What proportion of male and female progeny of F1 will have horns? Now, see, the concept of this question is very easy, okay? But if you do not know it, then you will like, what kind of मेल में और रिसेसिव है फीमेल में. वेल ऐसा होता है और इस पर्टिकुलर फिनोमेना को कहा जाता है सेक्स इन्फ्लुएंस्ड कैरेक्टर. ओके, इट इज कॉल्ड एज सेक्स इन्फ्लुएंस्ड characters. Okay, so what do we mean by sex influenced characters? Sex influenced characters are governed by the genes which are present on autosomes. Okay, so they are not present on sex chromosomes. The genes are present on autosomes. That means they will be equally present in all the gender, all the sex of the population. But the expression of that will vary from one gender to another. So in one gender, it is saying that it is behaving like a dominant allele. Okay, so what is the characteristic of a dominant allele? That even a single allele present will produce the phenotype. So if the male is in male, it is dominant, means having a single capital H here. Okay, other allele blank. Then also the male will be horned, will have horn. Okay, so that is for the male. What about the female? In female, it is recessive. Means in female, this H has to be present in homozygous condition only. Then only the horned female will be formed. Do you all agree to that interpretation, to this interpretation? Okay, now when you understood this, so now let's make the cross here. So cross is saying that a horned female is crossed with a hornless male. Okay, so horned female. So horned female means HH. Hornless male means H plus H plus. Okay, so HH is crossed with H plus H plus. Right, and then the F1 will be formed and one of the resulting F1 females will be crossed with hornless male. Okay, so I'll write it down the cross in a neat way here. So we have, this is female, okay, who is horned. So horned female is crossed with hornless male. Okay, next generation, you are getting only one genotype, that is HH plus. Okay, but because this is a unique situation, it's a sex influenced character, so the males and females will produce will show different phenotypes. Okay, so what is the difference here? That if it is a female, So females will be hornless because only one small h allele is present. Okay. But if this individual is male, then it will be horned because one allele is present here. Sorry, it will be horned. Okay. So this is hornless will be here and the males will be horned, right? Because that is the uniqueness of this H allele, which behaves like a recessive allele in female, but it behaves like a dominant allele in male. Okay. Now the next cross is what? That you take this female, the F1 female. Okay. That is HH plus is the female here. Okay. Character is hornless and is crossed with a hornless male again. This is also hornless. Okay. Now what do you expect in the next generation? Right. So for that, quickly we can make a Punnett square. So if I write down the gametes from the male, from the female here, so it is H and H plus. The male will produce only one type of gamete, that is H plus. So it will be H plus. This will be H plus H plus. Okay. Now in this situation, what we will have? So if it is, see this part who is H plus, okay, so it will be hornless only, right? But let's see this situation, what will happen in case of male and female. So for this genotype, if it is female, then also it is hornless. If it is male, then also it is hornless. Okay. Now what about this genotype? So again, we will split it into male and female. So if it is a female, okay, so in female, only one capital H is present, right? So that means it is not able to form the horn. So the female will be hornless. What about if it is male? In male, this H allele behaves like a dominant allele. So having a single allele also will make the male horn. Okay. Right. So now you see the males and females. So what do we see? That all the females are what? Hornless. Okay. So in the next generation, what will happen for the females? All will be hornless. And what about the males? In males, 50% will be hornless and 50% will be horned. Okay. So 50%, I'm writing HL for hornless, and 50% will have the horn. So this will be our conclusion. Right. So what will be the answer? That 50% of the male progeny will be horned, but none of the female progeny will be horned. So answer will be option 2. Okay, so we have a couple of more minutes and we will discuss one more question now. And I expect all of you to join our Zoom class. The link will be posted soon. Okay, stay here so that you can clear your one-on-one doubts. Another question regarding this kind of pathway. Okay, the pathway related question. So consider the following hypothetical pathway. What is the pathway given? Capital H allele converts X substance to H substance. X substance to H substance. Okay. Small h allele cannot convert X to H substance and leads to the phenotype O. Okay. So if this H is not present, okay, capital H is not present, in place small h is present, then what it will do? It will produce phenotype O. Capital A allele converts H to A, and small a allele cannot convert. Okay, if it is small a, conversion cannot happen. Capital A B allele does the conversion, but if it is small b allele, conversion cannot happen. Okay. Now an individual with capital A phenotype when crossed with that of capital B phenotype, have a progeny with O phenotype. Which one of the following crosses can lead to the above observation? Okay. So first of all, see. So first we have to write down the possible genotype of these two individuals who are crossed. Okay. So the individual is with A phenotype. Means what? That he has at least one capital A allele for sure. Other allele we do not know. Leave it blank. Okay. Now what do we have? Capital H. So now A to work on, you need the H substance. Okay. So if the individual is having the A phenotype, means यहाँ पे H substance तो बना ही होगा. और H substance अगर बना है, means this individual has at least one capital H allele. मैं दोबारा बोलती हूँ. देखो यहाँ पे अगर फिनोटाइप ए है, ओके, सो फिनोटाइप ए कैसे बन सकता है? जब कैपिटल ए अलील प्रेजेंट है. सो इस इंडिविजुअल के पास फॉर श्योर वन कैपिटल ए अलील तो है ही है. बट कैपिटल ए अलील जो एक्स सब्सटेंस है, जो हमारा प्रीकर्सर है, उस पे वर्क नहीं कर सकता. इट कैन ओनली वर्क ऑन द एच सब्सटेंस. ओके, सो इफ कैपिटल ए अलील हैज वर्कड, मींस this X has got converted into H. That means the individual also has one capital H allele. Okay, so this will be the general genotype of this first individual. Similarly, ठीक ऐसे ही जो B phenotype का individual है, उसका genotype भी यही होगा. Like instead of A, it will be B. So capital B allele एक होगा, so that the B phenotype is formed. But capital B allele can only work on H substance, means capital H is also there. Okay. Now the next generation, it is producing O phenotype. O phenotype कब होगा? O phenotype तभी होगा जब यहाँ पे स्मॉल एच अलील है, एंड स्मॉल एच अलील है तो ये पूरा कन्वर्जन ही नहीं होगा. ठीक है, और तब जाके phenotype O बनेगा. Means for H, what will be his genotype? For sure, we don't know about A and B, but for H, it has to be small h, small h. Okay, does not matter what his genotype is for A and B, but if it is small h, then for sure it will be phenotype O. Right? Now, यहाँ पे अगर हम देखेंगे, now check the options. Okay, see, option A, not possible. Okay, see, now this small h is here, right? So if it is small h means कहाँ से इसके पास आ रहा है? एक small h उसके इस parent से आ रहा है, एक small h इसके इस parent से आ रहा है, right? Means both the parents will be heterozygous for H. Okay, so you see option 1, not possible. Option 3, this is heterozygous, but this one is homozygous. Not possible. Here in option 4, this is homozygous, but this one is heterozygous. Not possible. Only in option 2 is the one where both H are heterozygous. Okay, so you don't even have to check for A and B. Okay, so if H is heterozygous, means O phenotype can be formed in this situation. So only one option has this. That's why answer will be option number 2. Is it understood, everyone? Okay, so now is the time for our Zoom class. I request the team, please put the Zoom class link in the chat box so that the students can join. And also it's the time for the freebies. So all of you who have attended here, you will get some freebies. The link will be posted here soon. So please check this link for the freebies, and the Zoom class link is coming right up. So just give me 2 minutes. I'll switch on to Zoom class, and please come here and we can do our one-on-one doubt solving session. Okay. And I have some extra questions. If time permit, we can solve that also in the Zoom class. Hope to see you all in the Zoom class. Thank you everyone for joining here. Thank you. See you all in another session. Please join the Zoom class and we can clear the one-on-one doubts. Thank you, everybody. Bye. Have a good evening.